present on inhibitors and cardiotoxicity. Dr. Dyas.
Good morning. As she mentioned, my name is Alexander Dyas. I'm one of the fourth-year residents over at Atrium Health Navicent in Macon, Georgia. And I really appreciate y'all's time this morning, allowing us to present our research. And I'll kind of jump right into things. My project was entitled PARP Inhibitors and Cardiotoxicity. And before I get kind of into it, I did want to mention my research mentor, Dr. Ilana Heffitz, who, without her help, this certainly would not have been possible. I have no disclosures to report, unfortunately. A little bit of background, PARP inhibitors are a type of targeted cancer therapy. We'll kind of get into the functioning of them here in just a second, but it's been utilized in ovarian, breast, prostate, as well as pancreatic cancers. And as we see kind of improvements in the molecular testing and as we get a little bit better at identifying these tumor genetics, there's a natural transition to a more targeted approach to cancer treatment. Our project specifically is a T0 translational study that utilizes a mouse model to evaluate an anti-cancer therapy. In our project specifically, we used olaparib as our representative PARP inhibitor, as it was the first PARP inhibitor that was approved by the FDA for maintenance monotherapy in ovarian cancer. So how do PARP inhibitors work? Well, they tend to be most effective in BRCA1 or 2 germline variants or tumors that are deficient in homologous recombination genes. The reason being, PARP is an enzyme present within the body that is functioning in single-stranded DNA repair. When you have tumor cells that are deficient in either BRCA1 or 2 or homologous recombination genes, they've naturally lost the ability to repair double-stranded DNA breaks. So as you see kind of in this image over here, the bottom side, when we combine the natural deficiency in these double-stranded DNA repair breaks, combined with PARP inhibitors, we effectively prevent the ability of these cells to propagate their DNA, and ultimately it results in cell death. Kind of jumping right into our project, the treatments we used, we took weight and age matched 10-week-old mice, and we treated them with either cisplatin, which was another representative anti-cancer treatment that has historically been used in ovarian cancer, and we gave either 1 milligram or 10 milligrams once a week for 3 weeks, or we treated them with olaparib, 50 milligrams per kilogram or 200 milligrams per kilogram once a day for 14 days. We also had a control group that was injected with just vehicle alone, and all of our treatments and controls were delivered via intraperitoneal injections. Ultimately, at sacrifice, the hearts, aortas, and retinas were harvested from these mice, as well as the serum in order to break down our analysis. So what we looked at was kind of a little bit of a variety of things. The first was a marker of vessel damage. So what we know is that cardiac fibrosis, collagen deposition, all of these play a role in cardiotoxicity. So in terms of macrovascular damage, the hearts were stained, they were sectioned, and the aortas were also stained and sectioned, and then wall thickness and collagen deposition were evaluated. In terms of the microvascular damage, we took the retinas from these mice and using a fairly well-established model to assess microvascular damage, evaluated the number of acellular capillaries that were present within these different tissues. We also did PCR analysis of 10 genes that were previously shown to be associated with cardiac fibrosis and cardiotoxicity. And finally, we took the serum from these mice and evaluated osteopontin, which is a matricellular protein that is involved in quite a variety of biological functions, but most importantly is the way that it's thought to increase and predict a poor prognosis for major adverse cardiovascular events. Our results, what we saw initially was a dose-dependent increase in cardiac fibrosis, so that macrovascular damage, in both mice that were treated with cisplatin as well as olaparib. We also saw a dose-dependent increase in collagen deposition in both olaparib and cisplatin, And I think that this image over here to the right is kind of one of the more interesting ones. This is our control group, and as you come down to the highest concentration of olaparib that we gave, you can see there's a lot of empty space, there's a lot of kind of room within that tissue, versus down here, there's virtually no additional space, everything is very crowded, and so that was kind of our marker of collagen deposition. We also saw higher doses of cisplatin and olaparib that significantly increased the aortic wall thickness. We noticed that there was a significant increase in microvascular damage as by way of those acellular capillaries. We found that there was a significant increase in the gene expression of several profibrotic genes within the heart. And finally, we saw a dose-dependent increase in the concentration of osteopontin within the mice serum following treatment with olaparib. So in conclusion, what we were able to draw from this study was that treatment with olaparib resulted in an increase in cardiotoxicity, specifically within our mouse model. And treatment with olaparib also increased the expression of several genes that have previously been shown to be associated with cardiotoxicity. And these are my references.
Thank you, Dr. Dyas. Bunjabal Ray, G1 Oncology, Medical College of Georgia. I have a question and a comment, so I'll ask my question first because it determines my comment. Were these mice, what was their germline BRCA status and what was their HRD status in your model?
So they were BRCA negative, and then they were not homologous recombination deficient.
Okay. Then in that case, my comment is that I would encourage you to replicate this study using niraparib instead, because niraparib, as you know, is FDA approved for germline negative HRD proficient, which would be your mouse model, and niraparib's primary side effect is hypertension. So I would be really interested to see what that looks like in this mouse model. If you're showing it with olaparib, which doesn't typically have hypertension associated with it, I think you'd see really profound results if you replicated it with the other drug.
Yeah, agreed.
Thank you.
Thank you.
Thank you, Dr. Dyas. All right, so